so here comes some fun stuff. Now we get to talk about our DNA and how our body uses DNA and the other things in and around the cell to build structure, to build stuff. Okay, in other words, and I don't like building stuff is sort of a simple term, so the term we should be using is to synthesize proteins. Okay, that's the fancier way of saying build stuff, right? To synthesize proteins. Okay, so we're going to start here with chromosomes, and we're going to work up to protein synthesis. This will be important, I promise you, because every step in this process requires a copying of information. Okay, and we're worried about radiation exposure interfering with this copying of information. So to understand how radiation can interfere with the copying of information, first we have to understand how the information is copied, right? How all this works. Okay, so let's do our best here. Every cell of your body, that's every somatic cell, will contain 23 chromosomes. And less importantly, 2 of those chromosomes are the sex chromosomes determining the gender or sex of the offspring. This will be like what we call nowadays the sex assigned at birth, right? Male or female. And 44 autosomes, which determine the traits of the organism. That's useful if you like. More importantly, 23 pairs. And very importantly, all the information necessary for the entire organism to develop is contained in the DNA of each cell. It makes sense, right? Because you took a sperm and an ovum, put them together, made one cell, right? That one cell had to copy everything to make 2 cells. Those 2 cells both had to copy everything to make 4 cells, right? So from the very beginning, when we were just one cell, that single cell had all of the information to build you, right? And every time the cell gets copied, all the information gets copied too. It's a full copy of the information every single time. Okay. In a little while, I have a video I told you that I'll play, and it shows the copying as best we can at full speed, and it's really cool because it goes really fast, and the scale and scope of it is pretty crazy how fast it's actually happening, and on the scale it's happening. And they're just showing you inside of one single piece of one single chromosome in one single cell, and it's pretty astonishing how fast this happens. It's amazing that we are here at all when you think about how fast and how complicated this whole operation is. Okay, anyways, enough of that. All right, so this picture is called a karyotype. This is basically a picture, a microscope picture of all 40 of all 46 chromosomes in the body. We're not going to do much with that except go, yes, there it is. There's the picture of all those chromosomes. There you can see that sort of when I describe them as like an X-shaped sausage, you kind of get what I'm saying, right? They're sort of like 2 side-by-side sausages. Roughly X-shaped if you stretch your imagination a little bit. Okay. All right. Let's zoom in now. So now we went from zooming way out to all the chromosomes all shown on a picture. Now we're going to zoom way in to just one single strand of DNA. Okay. So that brings us here. So DNA is a double helix twisted ladder. The helix part is the twist. Okay. The ladder part is that there's rails on the ladder, right? Vertical rails, and then there are rungs. Like the rung on the ladder is what you would climb. Okay. All the genetic information is in the rungs. Okay, the part that you would climb. All the genetic information is in the rungs, the horizontal stuff. The vertically running rails just hold everything in place. Now, if you are interested, the rails are made of sugar phosphate molecules. The rungs are made of built on nitrogen. They're nitrogenous bases. You may have heard me say, and I usually do it in the CPR when I tell you what the air is made of. I say the air is like 78% nitrogen, 21% oxygen, a little bit argon, a little bit CO2, a little bit other things. But I say, we use oxygen for respiration. And I say things like, we don't use nitrogen for respiration, but we do use nitrogen in other places in our body. Okay, one of those places is in the rungs of the DNA.
Well, the rungs
Rungs are the part you climb. Yeah. Rungs are the horizontal part with the letters. Yeah. So the rails run vertically. The rungs run horizontally. Okay. Rungs you would climb on. Okay. Importantly, the rungs of the ladder, the horizontal part, are, and this is what's really cool, all of this complex diversity of everything in the biologic world. This is why we think every living plant, human, animal, all of it has a common ancestor, right? A single-celled common ancestor from a few billion years ago, right? It's because we all share the same DNA, right? You share, I forget exactly, like, you know, you share like probably it's something like 99 point something percent of your DNA with like apes, but I was like bananas. You share a significant chunk of your DNA with like 90 something percent of your DNA. It's like 98% or something crazy, with like fruit and plants, right? So we're very similar to these other things at the genetic code level. Okay, all DNA is made of one of four, or combinations of one of four, what we call base pairs. Okay, so if you look at the rungs of the ladder, right, there's an A at the top on one side and a T adjacent to it. So A and T go together. Okay, then below that there's a G and a C. So G and C go together. Okay, A is for adenine. So you don't need to know like at the chemical level what these are. You just need to know that there's four different ones. These are their names, and we abbreviate by using the first letter of the name. Okay, so there's A for adenine. There's A up there, and there's T for thymine. T. Okay. Then you got the next rung down, G and C. G is guanine. C is cytosine. These are nitrogenous bases. These are molecules built off of nitrogen. That are our genetic code. Okay, so what's important with the DNA is that you look at one side of the rung, right? So if you take the rung on one side, like on the left, right, and you read from top to bottom, you have A, G, T. Okay, that on one side, the ordering of those letters will represent one gene. Okay. So we'll see in a second here how this is going to work. So the ordering of these letters on one side are your genes, okay? Those two sides are like a mirror copy of each other, right? And the rule is, and let me bring up the rule here, from side to side, T, thymine, can only bond with A, adenine, and G, guanine, can only bond with C, cytosine. So if you just looked at one side and it read AGT, right, on the left, AGT from top to bottom, those first three on the left side. If I only gave you that, you'd know the right side has to be TCA. Okay, so they're effectively a mirror copy of each other. Thousands of base molecules form the genetic code, organized in triplets called codons. Every set of three is called a codon. Yeah, big long strings of these, sets of three, so big long sets of three, so you take 3 and 3 and 3 and 3 and 3 and so on, you build a long string, okay, that will represent a specific amino acid. And if you know anything about protein, you know protein's built off of amino acids, okay? So, good, so each three on one side will represent the specific attachment to something called amino acids. We'll build up long strings in sets of 3 that will be a protein. Okay, so we'll get there, I promise you. So you've got like AGT, and then you've got C. I can't see the third one down. We'll go down here. Then you've got GTT, right? So you've got these sets of 3 base pairs that represent a specific amino acid, and you line up all the right amino acids in the right order, and you build a protein. Okay, so we need to be able to use this DNA to serve as a template to organize amino acids in the correct way to build proteins. That's what our body uses DNA for. So when we say it's the genetic code, or it houses all the information. This is what we mean, okay? The DNA is organized on one side of the ladder into sets of 3 bases, and each set of 3 will represent a specific kind of amino acid. You organize enough amino acids in the right way, and you build a protein, okay? So really what DNA is a template for building proteins. That's what DNA does. It's a template for building proteins. Okay. DNA is kind of smart about it, though. You can't build so just like you can't read a book when the book is closed, right? You have to open the book to read it, yeah? You can't do anything with the DNA when it's zipped up, okay? You have to unzip it to expose those bases, okay? Because those bases are connected, right? Like the A at the top is connected to the T on the other side. It's closed, okay? The G and the C are connected, right? All the way down this ladder, every one of those rungs are connected to the one adjacent to it, right? The book is closed, okay? In order to copy everything, we're going to have to open the book, right, and access each one of these, okay? So now you have some of the tools necessary to talk about protein synthesis. So here we go. This is going to get semi-complicated, semi-quick. Stop and ask questions, please. In the cell, when it's time, the DNA will unzip, okay? So I know do you see 3 separate strands on the screen? Yes. 3 separate strands? Okay. The top strand and the very bottom strand are one piece of DNA that has unzipped, okay? So you have the top strand, piece of DNA. The bottom strand is the adjacent side. They've unzipped each other now. Now, just focusing on the bottom strand, ignore the top strand for a moment, okay? On this bottom strand, it's now exposed, okay? So along comes, freely floating in the nucleus, just floating around minding their own business, what we call RNA nucleotides. These are effectively the opposite bases to this DNA's code, okay? These things are just sort of floating around. When they happen to like randomly bump into, like if this one happens to, if this one happens to randomly bump into this one and it fits, it'll lock in, okay? And if another one happens to randomly bump in and fit, it'll lock in here and here and here and here and here, okay? Building this long chain of something called pre-mRNA, or in other words, messenger RNA, mRNA, M for messenger, okay? So now, in the nucleus, a molecule of DNA unzips, exposing the code. The book opens up, okay? And along comes freely floating RNA molecules that attach to the exposed bases, forming a piece, a section of that, taking a section of that DNA and now basically making a mirrored copy of that section. So if this side of the DNA is a mirror of this, right? And this is a mirror of that, then this is an exact copy of that. Follow me?
How does the middle?
Because this is being synthesized. This is being created brand new, okay? This side is a mirror copy of that, right? And so only a mirror copy of this can be created here. So this is a copy of that. Okay, so the DNA it's copying in a mirrored way. Okay. Importantly, though, this and this strand of DNA will open up. It will allow to be copied. It will be allowed to be copied, creating this new strand, and then we don't care about it anymore. Okay, it's done now. Okay, it's done its job. At least for this point in time, it's done its job. What we care about now is this newly synthesized piece of mRNA, messenger RNA. Okay? That messenger RNA, so let's keep reading. That messenger RNA, the resulting sequence of base molecules on that strand, chemically mirrors a section of code from the DNA that we copied. It's just a mirrored copy of what we copied. This shorter mRNA molecule travels out of the nucleus. So let me give you an analogy, okay? I ask you to go into a library, find your favorite book, and I want to see it, right? But I say you can't take the book out of the library, okay? So what are you going to do? Well, you're going to have to figure out a way to get me the information that's in that book without taking the book out of the library, right? So one thing you could do is you could copy it, okay? And that's effectively what's happening here, okay? It's taking the book, copying it in the library, and bringing it outside of the library to me, okay? Maybe I only want to see chapter 1, too. Like the whole book would be like all your DNA, but I just want to see chapter 1, okay? So you go in the library, copy chapter 1, and bring it out to me, okay? So you had to open the book first. That's splitting the DNA, right? Then you had to copy down what was in that book. That's making the mRNA, okay? And then you get it outside of the library, okay? To add one extra layer of complication, it's more like you had to copy the book in a mirror, right?
Oh, backwards.
Yeah, I have to copy it backwards, right? So the mRNA strand is like a backwards copy of the book, okay, of chapter 1 in the book. Okay, so you have that backwards copy of chapter 1. You bring it out to me outside of the library, and I go, what is this? It's a backwards book, right? You'd like to show me a forwards book, yeah? Okay, so good. We've got something, though, right? We've got some information. We just got to, like, convert it back again, right? So this shorter mRNA molecule travels through rough endoplasmic reticulum out into the cell's cytoplasm where ribosomes are waiting, okay? Somebody's waiting outside of the library to help you now, okay? They're going to help you convert this mirrored copy of the book into a usable thing that I can now read, okay? So, assisted by ribosomes, nice little helpers, free-floating transfer RNA molecules, tRNA, are just, they're just hanging around, okay? They're hanging around outside the nucleus, and they will attach to the mRNA strand, and we're going to now form a protein, okay? So here's the mRNA strand you made, okay? This bottom strand here. This is the mirrored copy of the book that you brought out of the library, right? Okay. So now outside of the library, so the analogies only go so far as they go, right? So outside of the library, you take so it's a mirrored copy of the book, right? So in order to copy it correctly, what would you have to do? You have to hold it into a mirror, right? Okay. So you hold it into a mirror, right? And then you copy it again. And when you copy it again, you now have the chapter 1 facing the correct direction, right? Okay. So again, analogies only go so far as they go. So that's going to let us down a little bit once we get to the end of this here. But you get the idea, right? You took a mirrored copy of the book out of the library. You copied it again in a mirror, and you presented me with a correct copy of chapter 1. Okay. So that's what's effectively going to be happening here. So mRNA is your mirrored copy of the book. Alrighty? Now, again, the analogy is going to fail us here now, okay? So now, outside of the nucleus, you have freely floating tRNA. And these tRNA molecules, for interesting reasons that I won't go into explaining, are attached to amino acids. Amino acids, you can think of amino acids as like a Lego block that builds proteins. A Lego block that's like a piece of the protein, okay? Proteins are made out of amino acids. So you take these tRNA molecules that are sort of floating around with amino acids attached to them, and a specific amino acid will attach to only a specific spot on this mRNA, wherever it chemically matches, okay? And when enough of these line up on the mRNA molecule, assisted by this ball called a ribosome, you will line up a bunch of amino acids in a line, in the correct order, okay? This is copying the book in a mirror, again, copying your copy in a mirror, getting back to the correct version. All right, so the mRNA acts as a transporting machine for the code. The tRNA acts as a code translating machine, helping line up the amino acids in the right order. And the ribosome sort of just helps put things together. At the level that we need to describe it, that's all we need to know. The ribosomes help put things together. Here's the magic, though. At the end of all of it, once all the amino acids get lined up, assisted by the transfer RNA, that amino acid chain, that amino acid string, once it's fully lined up, will break loose of the mRNA, okay? And that amino acid string breaks loose, and there's your protein, okay? So this big long string of amino acids breaks off. That big long string of amino acids is the protein, okay? Now this might be an antibody, right? Antibody to some virus, okay? This could be any structural piece for the body. Doesn't matter which one it is right now, which is why we just drew them like little green squares, little green rectangles. But this is a piece of protein, a protein molecule, to be used somewhere in the body for some task. It can get very complicated when you ask which protein and how many and where's it going and that kind of stuff, right? But just think of like hemoglobin, right? Hemoglobin in your red blood cells. Literally the word hemoglobin means blood protein, right? The hemoglobin is produced in this way. Every one of your, the last number I remember, something like 15,000-ish different proteins we have in our body, and I bet that number's not exactly right, but there are thousands of different types of proteins that our body produces, okay? And each one of them, though, even though they're all different, is produced in this way, okay? And here's the cool thing, again, to remind us of this. Every instruction, every piece of every instruction to build every one of your proteins is in the DNA, which a full copy of that is in every one of your cells, okay? So in every one of your cells is all the information necessary to produce every protein your body needs, okay? So those are your genes. Your genes are the instructions to build proteins. Which proteins you build is up to the exact order that your genetic code, your genes are in, okay? And interestingly, some parts of the genetic code completely unused, okay? Other parts very, very used. Other parts are turned off, and then at certain points in life or with certain environmental stimuli get turned on, okay? Or due to certain hereditary information, they get activated, right? We have things called oncogenes, cancerous genes that switch on at certain points in life and under certain circumstances. So it's a big, complicated thing, but the idea is that you understand at least a little bit what our DNA is good for, right? Our DNA is good for providing the genetic code so that we can produce proteins for the body, okay? So if you change the genetic code, right, swap one of those base pairs out for a different one or completely just break a piece of the DNA off, right, you're now going to change the protein that you build, okay? Or you might not build a protein at all, right? So this makes for problems when you start to change the code, right? Okay, good. So let's finish the story. The story really ends importantly with making the protein, but here's what's neat. The rest of this stuff sort of gets recycled, okay? These transfer RNA molecules are going to break off and reattach themselves to freely floating amino acids because you ate a hamburger or something and you got a freely floating amino acids from the protein that you ate, right? So they're going to reattach to new amino acids. There you go. New amino acids get attached. And they're ready to be reused again. These are now ready to be reused and make a new protein. Yep. This mRNA strand is going to be dissolved by the cell of your body and completely reabsorbed and reused. So everything's repurposed, recycled. Okay. Let's do, oops, sorry. All right, hang in there with me for a little longer. Not as far as I wanted to get, but hang in there with for a little longer, and I will get you guys a break here shortly. Okay, good. So again, you're not supposed to be an expert at understanding protein synthesis. You're just supposed to understand that the genetic code, the DNA, offers a way to build the proteins that our cells need. Okay, build the structure our cells need. And the point is, if that gets changed, then you don't build the right proteins. You don't build the correct things that the cell needs to stay alive. Okay, so now let's move on. Let's talk about life cycle of a cell. So cell lifespans vary, but average is only 24 hours. A term for you, mitosis. Mitosis is the stage or the phase of the cell's life in which it splits. Phase of active division. Yes. Cells split by copying everything and then splitting down the middle and sending, a full copy to one side and a full copy to the other side. Now, mitosis is split into 4 phases. But if you notice here, take a look at this little pie chart, right? Mitosis is just this piece right at the top of the pie. Okay, these 4 tiny, skinny little slices right at the top of the pie, those 4 slices make up all of mitosis. Most of the cell's life is called interphase. Interphase is the time between cell divisions. So a cell divides. It's now a brand new daughter cell. It's going to live its life until it divides again. And we call that interphase. So interphase is the time between cell divisions.
Time between cell division?
Yeah, so interphase is all of the time, everything that's happening between cell division. Now, interphase itself gets split up into 3 things called gap 1, synthesis, and gap 2. It's just going to be less important than the mitotic phases, but you should know that this process interphase, this time of interphase, gets split up into these gap 1, synthesis, and gap 2 stages. Okay, let's say some important things now. So during interphase, chromosomes appear only as a diffuse granular mass of loosely coiled chromatin. Chromatin is the name for a strand of DNA, before like a packaged strand of DNA. So a series of bead-like globs of protein called histones around which the DNA molecule wraps itself before extending to the next bead. This is effectively a DNA fiber. So interphase, remember interphase is the time between mitosis. The chromosomes are basically unpackaged. Okay, they're all loosely spread out. If you looked at them with a microscope during interphase, most of the cell's life, right, you would look at the nucleus and think really, there's nothing really there, right? There's no like real structure, okay? You only can see the chromosomes during a small period of time during these mitosis stages, okay, when everything's sort of packaged up, ready to split. We'll talk about that, I promise. All right, so terms for us. Chromatin is a series of globs of protein called histones with DNA wrapped around it. You'll see a picture in a moment. Chromatin fiber, strands of twisted chromatin twisted together into a rope-like structure. We're talking nanometers across, billionths of a meter in size, right? Very, very small, okay? But, so when we show you that, right, know that we're, a single chromosome is housed inside of the nucleus of a single cell, right? You need microscopes to see even a single cell, right? So we're talking down very, very small as far as size scales go. So this little picture here shows you from unpackaged all the way through packaging, all the way through now to a chromosome. We're sort of zooming in and starting very zoomed in, and you start zooming out as you move through this picture, okay? So at the top left of this picture, what I've zoomed in on right now shows you the DNA double helix. So there's your twisted ladder with your nitrogenous bases, the rungs of the ladder. Cool? So you take those, and the DNA, the way our DNA works is to keep everything organized. Those that double helix DNA strand is wrapped around something called a histone. Histones are little globs of protein that basically help us package DNA. So that way it's not going to get all tangled up and messed up and break and that kind of thing, okay? So histone beads, which are little globs of proteins. They look like nice little round balls here. They're not exactly. But what'll happen is the DNA will wrap itself twice around a histone bead, and then the DNA strand will extend down and wrap around another one, and extend down and wrap around another one, okay? One bead at a time. The DNA wrapped around those histone beads is called chromatin. That strand created, little like a pearl necklace-like strand that's created, it's called chromatin. If you package enough chromatin together, you get a longer, it's like taking a piece of string, right? Several pieces of string and then weaving them together to make a rope, right? Multiple pieces of string make a bigger rope, right? Multiple pieces of chromatin twisted together make a chromatin fiber, a larger fiber. That larger fiber, if you follow the picture down, packages up into that sausage structure called a chromatid. So you have chromatin, packages up into a chromatid, okay? That chromatid is 1/2 of a chromosome, okay? So you get it all packaged up, you make a sausage and another sausage next to it. They're connected in the middle. That makes an X sort of shape, and that's your chromosome.
So the centroid and the chromos is DNA.
That whole thing is the DNA, yeah, all packaged up.
Whole thing.
Yep, the whole thing. We're just starting very zoomed in, and we're zooming out as we go down the picture.
Well, these little squiggly lines,
Mhm.
what are they?
Those are,
Here.
so this, this is zoomed in, and we're zooming out now. So all of this here, and all these squiggly lines here, is this,
Oh, okay.
right? We're just taking this, and we're zooming out, and that's it there. It's all packaged up, squiggled up there.
Okay.
That is this,
Yes.
this is that, and that is that.
Got it.
Yeah.
Yep.
Good.
Okay, cool.
Good. Fun fact about the word chromosomes. You guys know what the combining form chromo means? Color and some? Yeah, so put it together, colored body, right? I forget, it was Watson and Crick. Anyways, the scientists that discovered chromosomes looked at them under a microscope and injected some dye into the nucleus of a cell at the right time in the cell's life and went, huh, these little things took up the dye. There's little colored things in the nucleus of the cell. They go, well, what are we going to call a colored thing? Chromosome. It's not very smart, right? The word chromosome sounds smart, but it was just these scientists saying, well, I see these little things that are colored in the nucleus, so let's call them a colored thing. And then they just applied the medical terminology to scientific terminology to colored thing, and chromosome is the translation of colored thing. Yeah, so chromosome just means little colored thing. Yeah. Okay, good. So there we go. We have a reason why we have the word chromosome. Bless you. All right, so there you go. You have a complete chromosome is a double chromatid structure, an X-shaped double chromatid structure. After synthesis, we have the double chromatid. So B happens after the synthesis phase. Okay, so now we have basically it's ready to copy now, ready to be split apart. It's already copied, ready to be split apart by the time we get to stage B when it looks like an X, and we can see it under a microscope. So let's do this. Let's review the life cycle of the cell in the mitosis stage right now. Okay, so mitosis is important. Mitosis, also called replication division, because it results in a full identical complement of genetic material in each new daughter cell. So unlike, people, right, larger organisms like us mammals, right, when we reproduce, we still exist, right? We make two people make a new organism, we're still here, right? But when a single cell splits, you can't say, well, which one's the original cell, right? Because neither one is the original cell, and both of them are, right? It's a single cell splitting into two, okay, copying everything and splitting into two. These are two new cells. The original one's effectively gone. Okay, it's part of both of these. Okay, and so the single cell goes to two, and we call them daughter cells. Right, the parent cell doesn't really exist on its own anymore. It is just part of each of the two new daughter cells. Okay, mitosis divides into 4 stages. These are called prophase, metaphase, anaphase, telophase. And we'll just give a brief explanation of each. And you'll see in which one the chromosomes become most visible. Or if you don't see, I'll tell you. All right, so here you go. Stages of mitosis shown. Do I have? Oh, yeah, okay, good. All right, so A, B, C, D, 4 phases of mitosis. Prophase. The nucleus swells. The DNA is not, it's copied. It is not ready to divide yet. Okay, so you see A is shown, that's prophase.
So it's getting ready to split?
Yeah, it's getting ready. All of these are getting ready to split. A, B, C, at least A and B at a minimum are the cell getting ready to split. C and D are the cell actually splitting. So the prophase, the nucleus swells in preparation for the split. Here's the important one. Metaphase, the nucleus elongates. The chromosomes line up along the equator, right in the middle. B. Metaphase. The chromosomes line up, and here's what's cool: tiny little microtubules will attach to each side of each chromosome on each side and get ready to pull it to opposite sides of the cell. Okay, so metaphase is where all the chromosomes line themselves up along the middle of the cell, ready to be split. Okay. Only during metaphase do the chromatin fibers condense so much that the chromosomes become visible under a microscope. That means our scientists that discovered chromosomes did it during metaphase. Yeah, that's what I thought. Their names, by the way, I didn't want to... So I have too many names bouncing around in my head. I was thinking Watson and Crick, and I was right. So the two scientists' names were Watson and Crick. If that's useful at all for you, you won't be tested on it, but it might be a useful thing to remember. So those are the two guys, okay? And they're the ones that when the cell was in metaphase, they went down, there are little colored things right there, okay? And they called them chromosomes. All right, good.
So when they're in the metaphase, you can visually see.
Yeah, not like naked eye, but like through a microscope, yeah.
Okay.
And the rest of the time, they're either unpackaged or too split up to be visible.
Okay.
Yeah,
Under the microscope.
under a microscope. Yeah, they just you can't see them under a microscope without dyeing them,
Okay.
and the chromosomes won't take up a dye until they're all like sort of smushed all together right during metaphase. Yeah. Okay. Let's go C and D. C and D is the actual splitting. C is called anaphase. The double chromosomes split and are pulled to opposite ends of the nucleus. C, anaphase. D, telophase. The new cell membrane divides the cell. The nuclear membrane dissolves and will reform, and the cell splits. Half goes to one side, half goes to the other, and then the thing just kind of pinches off in the middle, closing off, making two new cells. After that, the cell is now an interphase. In other words, it's living its life. It's got, on average 24 hours to live its life. Unless you're a neuron and you're amitotic and you never do this, right? That's why nerve cells are special. One reason why they're special. But on average, every cell in your body is living its entire life in 24 hours. So it gets to the end of telophase, and it's got 24 hours till it's back there again.
What do you mean? That cell doesn't die like once it's.
So, okay, so you go to telophase, you make two new cells, right?
Yes.
On the average, each of those cells is going to wait 24 hours and then split again.
Okay.
Yeah. Now, sometimes the cell just dies, right? But if the cell's going to keep going, it's going to, on average, split again in 24 hours.
Okay. So it like never ends.
Well, it only does this a certain number of times. So the DNA in each cell can only be copied a certain number of times before it starts to get, I'll say messed up when it copies. Yeah. Until the copying starts to get messed up, and then the cell's just going to build some random nonsense, and that random nonsense is not going to keep the cell alive. The new cell's alive, and it'll die. It might not even make it all the way through the mitosis phase. So, yeah, at a certain point, the cell can't stay alive and can't keep dividing. Yeah. It's like like the analogy would be like, you take something, you want to make a copy of it in a copier, like a literal copying machine, right? And you put your paper down on the copier, you make a copy, and the copy you get out of the copier is never as good, right? Okay, so then like take the copy and copy that, right? And the quality degrades again, right? And you copy again the copy, and you take a copy of that copy, and eventually what you end up with is just a terrible, grainy, granulated, degraded version of what you originally started with, right? It's kind of like that for cells, right? They can only copy so many times before it starts to make some bad mistakes, right? Some bad, mutations. The buzzword for this is called telomeres. Telomeres are the names for the very, very tips. Like a shoelace, right? Has the shoelace? Yeah. Little plastic. If you're wearing shoes with laces, little plastic at the end of your shoelaces, right? That's like a telomere for your DNA. Okay. Like when that little plastic tip wears out, what happens to your shoelace? Sort of like unravels, right? Same thing happens to the DNA. Okay. So think about your shoelace, right? Take your shoelace and like copy it, right? Each time you copy the shoelace, that little plastic tip gets shorter and shorter and shorter. Eventually, it's gone forever, right? And at that point, the cell's little shoelace end will be gone, and the DNA will sort of unravel and won't be able to be copied anymore. Yeah, that's about as hand wavy as I can give an explanation of why cells have a limited lifespan. Interestingly, in certain animals, scientists have been able to extend the telomeres in them, meaning that they can get animals to regenerate youth and live effectively indefinitely. Yeah. Now you can't haven't applied it. I don't think haven't applied it to people, but I believe they've done it in mice and other small animals. So kind of cool, right? Yeah, we can solve.